Hello everyone, I'm Charles Mulligan. I'm Deputy Director of the St Jude Comprehensive Cancer Centre, and it's my great pleasure to welcome you to the opening lecture of the Fall 22 Science of Childhood Cancer Seminar Series. As many of you would know, the series began as a way to continue scientific exchange during the pandemic, and it's been very successful. We've had over 5,000 people register, with several hundred people joining for each seminar, and this now starts our fifth series. As housekeeping, during the seminar, if you have questions, please feel free to submit those. Use the chat function of WebEx to enter them and address them to all panelists, and I'll moderate questions at the end of the lecture. Our speaker will also stay for an extended discussion at the end of the seminar series, so after the top of the hour, so please consider staying for that. So with that, I'm truly delighted to welcome our opening speaker for this series, Professor Carl Jude. Professor Jude holds the Richard W. Vague Chair in Immunotherapy and is the Director of the Center for Cellular Immunotherapies and Director of the Parker Institute for Cancer Immunotherapy in the Perelman School of Medicine at the University of Pennsylvania. He had graduate training in immunology and malaria with Dr. Paul-Henri Lambert at the World Health Organization in Switzerland, and postdoctoral training in transplantation biology with Don Thomas and John Hansen at the Fred Hutchinson Cancer Research Center in Seattle. He's board certified in internal medicine and medical oncology. His lab studies mechanisms of lymphocyte activation as related to immune tolerance and adoptive immunotherapy for cancer and chronic infection. In 2011, he published landmark results detailing a new therapy in which patients with chronic lymphocytic leukemia were treated with genetically engineered versions of their own T cells. Of course, that is CAR T cell therapy. And this revolutionary therapy was granted breakthrough therapy status by the FDA for acute leukemia and subsequently diffuse large cell lymphoma. Professor Jude has published over 500 manuscripts and has been awarded many leading prizes and honors, just several of which are election to the Institute of Medicine, the National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. The Paul Ehrlich and Ludwig Darmstädter Prize, the Novartis Prize in Immunology, the Karl Landsteiner Memorial Award, and a Lifetime Achievement Award from the Leukemia and Lymphoma Society. So Professor Jude, it's our privilege to have you here today. Welcome.
Thanks so much, Charles. It's a real pleasure to be able to join you all virtually, for this series. And I have to say, probably, you wouldn't get me to Memphis in the middle of August, given the humidity there. But it's great to do this virtually. But I do wish I could catch up with a great group of basic and translational scientists that you all have, at St. Jude. So I have a sort of a smorgasbord of our CAR T cell results that I've selected. A lot of it's either recently or unpublished, and so I hope we can have some interesting questions later. My disclosures are here, and so what I'm really talking about, and those of you who are working with CAR T cells understand, is this evolving area of synthetic biology where we can try to make T cells and the immune system in general more potent against cancer. And in this review I wrote with Michelle Sadelain a few years ago, we're faced at this point with alpha beta T cells that express either a transgenic T cell receptor presented on HLA polymorphic molecules, or CAR T cells, which target the surface of cancer cells and are not HLA restricted and have their own pre-programmed co-stimulation. Both of these are very potent and I think have non-overlapping uses. And I just wanted to bring this out, this idea that, as now that we can sequence tumors, as Charles has shown us, find the cause exquisitely of leukemia, you would think you could fix it, as Cicero said. And then, I moved to Penn in 23 years ago and Peter Knoll was my idol. He had discovered the Philadelphia chromosome and, the first genetic cause of leukemia. And It took 40 years until Brian Drucker and Charles Sawyer's, had imatinib, and it got FDA approved. So it didn't happen so fast as Israel would have predicted. What I'll tell you today, I think, is that the cell and gene therapies, now that we have the tools, are going to be much more rapidly developed. And I think that's a really exciting concept that the drug development time now, when you have uh, uh, tested and validated systems like gene modified cells, I think is going to be a rapidly developing platform. So, we got into this in 2010 infusing patients with chronic lymphocytic refractory leukemia, CLL, in 2010, and then treated a variety of other diseases, and then beginning in 2012, pediatric pre-B cell ALL, all with autologous T cells that had a CD19 CAR targeting moiety, and leading to FDA approval in 2017. And so now we have two FDA approved, actually three now, CAR T cells targeting CD19 and also now BCMA. They're similar but different in details. As shown here, the vectors, lentivirus versus gamma retrovirus, and the promoters, et cetera, signaling domains are different. And really strikingly, the persistence in patients with leukemia is quite different, with the 4BB based CAR surviving much longer in patients than in the CD28 signaling. However, the response rate and apparent cure rate in lymphoma is identical. So long-term persistence isn't required, at least in lymphoma. The data, I think, is in leukemia, unless you have a crossover therapy, such as a salvage transplant. Longer persistence is better for preventing resistance. So we began evaluating signaling domains back in early 2000 when Marcella Maus was a graduate student in my lab, and she showed this data here, that you could get log linear proliferation of CAR T cells, of actually just primary CD8 human alpha beta T cells, by repeated stimulation, as shown by the arrows with 4BB ligand and CD28. And in the lower right, you could see that with the technology we had back with microarrays, then this proliferation was polyclonal, as judged by the CDR3s. And so there's many different signaling domains in testing. We have the FDA approved ones with Yescarta and Kymriah. And Dario Campana then at St. Jude had worked, and he collaborated with me on developing the initial preclinical CAR against CD19. But there's many other signaling domains, including some that work through NK signaling molecules like DAP12 and TLR signaling, such as MyD88, that I know some of you at St. Jude have been developing. So we had now this I think great time during this last year, where we had a 10-year follow-up from the initial patients treated. And on the left are the first two adults we treated with refractory CLL, and they had 10 years of both of them of disease-free survival, leukemia-free survival. And then Emily Whitehead in April also now has 10 years of survival. So we could finally say, I think, in these early treated patients, that they're cured based on the 10-year leukemia-free remission. And Joost Mellenhorst has studied the initial CLL patients, and we're doing this now with Emily, with Steve Grupp's group at Children's Hospital. But what Joost found, surprisingly, was in the CLL patients, that long-term, when staining with an anti-idiotype, that they first of all still had CAR T cells. About 1.5% of their peripheral blood T cells expressed the idiotype, and it was more CD4 cells than CD8, which was not how we had infused the patients. Back in 2010, when they were infused, it was more CD8 cells than CD4 cells. So the long-term surviving CAR T cells were CD4. And on this single cell analysis, we found with UMAP projections that we could find both CD4 and CD8 cells. So for instance, CD4 cells appeared activated. They have Ki67. And we also found some double negative cells, which, as I'll come back to, are actually gamma delta T cells. But at the infusion, and early on, this shows at 2.5 years and 1.8 years for the first two patients. They had multiple kinds of T cells as shown by the UMAP projections. But as they progressed, the surviving cells and persisting cells in these patients became essentially CD4 cells, as I mentioned, that are Ki67. And over here in panel D, we've stained either in their peripheral blood, the looked at the cells that are CAR positive and CAR negative, and they're activated cells. They have whereas their endogenous cells that aren't CAR cells are Ki67 negative. They're in G0, whereas these other cells have activation. And actually, we found on single cell RNA-seq they have cell cycle progression genes that you see in S and G2 and M. And the site-seq data that we did with investigators at Children's Hospital shows both in green the protein expression. So this is T cell receptor alpha beta and gamma delta staining. We found in this patient 3 months after staining that the dominant cell, surprisingly, was a gamma delta CAR T cell in the patient. And as I mentioned, they have effector molecules like granzyme. So they evolved in the patients. Initially, they were alpha beta CD8 cells, then gamma delta, and then long-term CD4 cells in this patient. So the cells evolved, and over a decade in these CLL patients while in remission. They had long-term B-cell aplasia, and unlike endogenous T cells, they maintain persistence with, I think, what's tick over proliferation, as measured as we saw in single cell RNA-seq, and that's likely from the reservoir coming out from hematopoietic stem cells of CD19 lineage appearing and then being whacked by the CAR T cells. That's probably what sustains them in the absence of any detectable leukemia. So CAR T cells can maintain long-term remission. More than 15,000 patients have now been treated. If you add them up from CD19 and BCMA, and really importantly for the field, no cases of T cell transformation have occurred with autologous CAR T cells. There are 2 cases reported of transformation with allogeneic T cells using Sleeping Beauty in a very specific situation that we could discuss that occurred in Australia in blood and published in Blood last year. And in fact, I think Steve Gottschalk has commented on that. This is a very important thing for the field, but the autologous cells have been saved in both pediatric and adult patients. Major effort is underway to shorten the vein-to-vein time, automate it, and then as I'll be focusing to incorporate multiplex human genome editing into these CAR T cells. So the first, pediatric patient we treated, and this is Steve Grubb's patient, was treated in 2012, and she had a very severe cytokine release syndrome, as you can see with interleukin-2 and IL-6 in the serum. And she's now 10 years out, but she nearly died from cytokine release syndrome. In 2015, she was able to meet President Obama during the initiative, the Precision Medicine Initiative that Francis Collins announced. And this is how she got out of school that day, from a handwritten note from Obama that she was excused to be there on a Monday. But this is what happened in her. We didn't see this acute rapid proliferation of CAR T cells in the CLL patients like we did in pre-B ALL pediatric patients. So this patient, Emily had, even 6 days after we treated her, still a bone marrow packed with CD19 and 20 positive blasts. but just 14 days later, she had uh, um, a deep remission that's been maintained, and many bone marrow now packed with CAR T cells. When you do the metabolic um, bookkeeping here and you count that the liter, that in a 7-year-old, there's more than a liter of bone marrow, and what was in her blood, she had more than a kilogram of CAR T cells that she made biosynthetically in her within 3 weeks after infusion. So it's a massive metabolic challenge that the patients actually become bioreactors. And this is her fever course that we published. She had a biphasic fever. Some of this is she had steroids started. She had fevers almost to 41 degrees. That was not due to infection, was cytokine mediated. And it's only when we gave her tocilizumab that the fever abated and she actually, the immunopathology repaired itself. And so that led to tocilizumab, anti-IL-6 receptor, becoming co-labeled for this. And it's now also being given for myeloma patients with BCMA CAR T cells, which can also trigger cytokine release syndrome that clinically is indistinguishable from a CD19 CAR T cell. And all that was serendipity. So my daughter, Sarah, is shown here. She had JRA, which is now called juvenile idiopathic arthritis, diagnosed in 2001. And in 2010, I was a president at that point of the Clinical Immunology Society, and I gave the presidential award to Tada Kishimoto, who had invented tocilizumab. And then, in 2012, Emily Whitehead, the reason we gave her that drug was because as an arthritis drug, we tried it for CRS because I knew about it just from my daughter. So this is a little bit of a note for the junior people on this, the postdocs and graduate students. Serendipity matters. And I showed here three different, when I was a medical student to an assistant professor and a tenure professor, the most recent one being here, that's the Emily Whitehead thing, that she would have died if it hadn't been lucky that I happened to know about that drug. And these other papers also occurred because of serendipity. So you'll find unexpected events in your career and finding, and if those are often more interesting than the ones that you do as a hypothesis-driven experiment. Okay, so we were able to follow these CAR T cells, and in your Goodman and Gilman, it would say that we would follow PK of new drugs with ADME, the absorption, distribution, metabolism, and excretion. But Goodman and Gilman doesn't talk about living drugs like we have with CAR T cells. And so we've now been able to analyze these in patients with, in this case, the cohort of 90 patients with the refractory pre-B cell ALL, adults and kids. And the expansion phase here of the CAR T cells in the blood is about 0.7 days, the initial doubling rate. And then this contraction phase here is around 4 days on average. And then some patients lose engraftment and others have basically indefinite, as I showed, 10-year engraftment with on a beta phase today. And we've looked at modeling of this, compartmental modeling done by pharmacologists at Penn and Novartis, and unable to find any dose response on the PK. The Cmax is dependent on tumor burden, and we find more cytokine release syndrome with a higher Cmax. So it's not related to the dose, unlike with standard drugs that get metabolized. Here, target causes proliferation in B cell tumors at least. And then they have some transition rate constants into memory CAR T cells. And then they also have plasticity, as I've shown, and I'll discuss more about that. So CAR T cells are living drugs. I think that's one of their major strengths, is that they can work as an immune system does. And the pharmacology is very different from inert, non-dividing drugs. And the safety of autologous CAR T cells has, I think, been established. as I've mentioned. And the main new medical side effect observed with CAR T cell cytokine. So cytokine release syndrome, we've talked about the other major issue and the black box warning that you'll see with Kymriah is the ICANS or neurologic toxicity, which often follows. And also, unbeknownst to many people, the package insert for Kymriah includes that it can cause a false positive assay by PCR for HIV, and that's due to the backbone vector sequences from the HIV-based lentiviral vector. But the neurologic toxicity has not been so obvious, the cause of that. In studies we did with Howard Young and Susa Pathy, we found unexpected CD18 in the brain mural cells, the pericytes in the brain. And we published that in Cell last year. And we've just recently published with David Tichy at Children's Hospital a proteomic investigation comparing cytokine release syndrome and ICANS. And so the data set is 26 patients that were treated, and then we did retrospective chart analysis to identify, the 26 patients who had severe or minimal CRS, and then whether or not they had neurotoxicity or ICANS, and 12 patients have that. And what David Tichy did was use this platform called Olink from Sweden to look at 1,400 analytes in the serum of these patients. So longitudinally followed, they were independently, the patients judged on their CRS and ICANS. And this has just recently came online on the details of this work from David and that lab. And so many differentially expressed genes were found at the time of peak cytokine release syndrome. Not surprisingly, IL-6 was off the charts as far as elevation, another IL-1 pathway like IL-1 receptor, and but also other cytokines like IL-2. And and then if we looked at mapping, here the full enrichment of cytokine pathway on a gene set enrichment analysis was the most identified. And the most surprising aspect of this trial, is patients were followed and they had either cytokine release syndrome or de novo ICANS or CRS evolving into ICANS. So CRS is shown in the dark lines here, and what you can see is the patients who had CRS with ICANS had prominent elevations of IL-1, alpha, beta, and then if they transitioned from CRS and dark to ICANS, they got IL-18 in the serum. And only one patient with CRS only without ICANS had IL-18 in the serum. So we found in this initial discovery set that IL-1 and IL-6 proteomically are what we see, which many people have confirmed. But for ICANS, IL-18 and some other markers show up that appear to be distinct from what's seen proteomically in CRS. So that you can see the details of that, but there's a trypsin-like receptor that also comes up with ILS with ICANS, chymotrypsin elastase receptor 3, and also mast cell Ig-like receptor 1 came up as a predictive biomarker for CRS. So this needs to be validated with other data sets, but it looks really intriguingly that the ICANS probably has different pathophysiology than CRS. It's not just spillover into the blood-brain barrier of CRS. So the initial trial we did with David Porter on CLL patients had a 57% overall response rate with CR and PR patients, but also non-responding patients. And what we found there was that the PK really correlated quite well. This is looking at a year after infusion on a log linear scale, and that there was much higher peak PK in the patients with CRs and PRs compared to non-responding patients who either had poor proliferation or lost engraftment entirely. But we also found that the time to PK of the Cmax and PK varied from 7 to 10 days, as shown here, to as late as 50 days, as shown in patient 10. And Joe Faretta looked at that in great detail, a CLL patient who also had a well-differentiated lymphoma, it's a B-cell tumor in the blood and in the lymph nodes. And the patient had delayed CRS as of 70 days from a first infusion of CAR T cells. This is, again, cytokines here in the serum. The different disappearance of blast cells was delayed, but he had then a stringent CR. And had a delayed disappearance of these, in this case, retroperitoneal adenopathies. And what Joe found was that this was massive expansion of clonal CAR T cells, unlike our other patients, where it was polyclonal at the T cell receptor level. And in this case, it was 2 months after infusion at the time of peak CAR T cell Cmax, it was CD8 alpha beta T cell that predominated and it had a V beta 5.1 TCR. And it went from a lot of over 1,000 insertion sites in the infusion product at this time of Cmax of the CAR T cells to only having one insertion site, and that was in the TET2 gene on chromosome 4, Q24. And what Joe found, along with Rick Bushman here at Penn, was that the CAR had inserted intronically between exons 9 and 10, and that led to disruption by NHEJ, and then so the exon 11, where the dioxygenase of TET2 is expressed normally, was not expressed on that allele where he had insertion. So that showed that those cells could outperform, at least by proliferation, the wild type cells that have wild type TET2 in that patient. And Joe has since gone on to show that you can do this intentionally now using CRISPR knock-in approaches or base editing and get by knockdown or knockout of TET2 enhanced performance of alpha beta T cells. And this really shows a profound way to study by reverse genetics the CAR T cells in patients, comparing those to what's infused in the patient. And leading to this Yogi Bear idea is when he was on the Mets, that and a coach and a manager, you can observe a lot just by watching. And he also said, if you come to a fork in the road, take it. I think really the ability, since we have genetically modified T cells to learn about the immune system is an amazing opportunity now studying our patients after infusion. So we intentionally modified the genome in a pilot study reported in Science last year. And this was just to test, could we do polygenic multiplex editing with ex vivo manipulated CD8 alpha beta T cells. And these were in 2 patients with sarcoma and 2 with myeloma targeting with a TCR targeting the New York ESO1. TCR was a study led by Ed Statmauer. And then the bottom line with that study was using the technology that was available with CRISPR-Cas9, so standard Cas9 and not a high-performance Cas9 nuclease. We could get digenic and trigenic editing at the level of a single human genome. So this is here on the left panel, 27 cells were analyzed with the 10x genomic pipeline. And these are cells that in the infusion product also had an integrated lentiviral vector for the New York ESO1 T cell receptor. And Some cells had monogenic editing, like in this Boolean Venn diagrams. I mean, only 11 different, or 11 NHEJs to knock out that gene, but 6 cells had all genes knocked out, the PD-1 gene and the T cell receptor beta chain and the T cell receptor alpha chain. And now with more modern, as I'll show, CRISPR-Cas9, we can do this at 100% or not, over 90% efficiency in patients. So I think this really bodes well for making synthetically enhanced T cells, and where we really need it the most is in solid tumors. And this data from Dan Chen and Ira Melman showing the so-called cancer wheel and the many ways of and aspects that have to happen when you trigger an immune response against cancer. And what we can do with adoptive transfer is enhance this steps 6 to 7 with adding in CTLs and CAR T cells. And this needs to overcome the many immunosuppressive effects in the solid tumor microenvironment that are much more pronounced than in most bone marrow-derived malignancies. So we have a number of trials at Penn that are open, testing various aspects. What I'll just show some data about our mesothelin CAR T cells, which are primarily used for ovarian mesothelioma and pancreatic cancer. And we have a lot of studies that are on the launching pad as well. But recently we've studied a model of and developed a model in vitro to look at the dysfunction that occurs in T cells and in the solid tumor microenvironment. So this is a model that Angela Asnar in my lab with Charlie Good and Shun Kurimitsu developed, adding a KRAS pancreatic tumor line called ASPC1 that has mesothelin expression. So we're using a mesothelin CAR that has a 4BB signaling domain, and we add and flooded the culture system with tumor cells so that the CAR T cells never are able to eradicate the tumor. So trying to mimic the E to T ratio we would see in humans with metastatic tumor burdens. And then studying the CAR T cells as they evolve over time. And the details are reported in this paper in Cell last year. And what we found was that with continuous antigen exposure, this so-called stress test here of adding repeated cancer cells that are KRAS transformed, that we found progressive CAR T cell dysregulation and plasticity with an NK-like T cell transition in vitro. And we found transcription factors induced that are not normally in alpha beta T cells, in particular ID3 and SOX4. And so we've now studied that more in some patients. And this is data that Mark O'Hara, a clinician at Penn, has from patients treated with metastatic pancreatic ductal adenocarcinoma. So these patients all have liver mets. They're given CAR T cells either regionally into peritoneal fluid or with a microcatheter into liver metastases. And so these are the patient ID numbers. And what we can find is that there's a reduction in the cancer cells. This patient 57, for instance, had peritoneal fluid, but there's not much persistence. This is the CD45 positive CAR T cells in the peritoneal fluid. And in patient 59, who had the catheter and then a subsequent liver biopsy of a metastatic lesion, had initially, this is the most intriguing, had mesothelin positive, EpCAM positive, double positive cancer cells. And at day 14, he did not, although later the tumor regrew. So there's some anti-tumor effects. They're not RECIST responses. But we do see in these patients the dysfunction that I mentioned in our in vitro model. So we see induction of checkpoints in the CAR positive cells in these biopsies. So this is PD-1 and CTLA-4. We see TIGIT induced, and then ID3 and SOX4 that were reported in the SOX in the cell paper in vitro. So this is on cells re-obtained after a biopsy from patients. They have markers of NK cells, and which is a hallmark of this dysfunction that we find with solid tumors. So I think a major area, and I think many would agree now, is how do we edit cells to improve them and prolong their function in the patients with solid tumors that Stanford Health's lab has this nice review on the many aspects that you will find. And you'll find this also in many sarcomas. And we have some data that I'll just show some sound bites here of unpublished data. So I mentioned ID3 and SOX4, and this isn't in the cell paper. Angela has now gone ahead and treated mice, and we, in this case, done single knockouts of ID3 or SOX4. She's also done double knockouts now. But the ASPC1 tumor that grows and kills NSG mice, these are large tumors here treated with a single CAR T cell injection here. Much better response if the CAR T cells have knocked out SOX4 or ID3 transcription factors. And this just shows that the tumor volume is much less. So this is one exciting approach, is a cell intrinsic modification of the CAR T cells of these transcription factors that are in healthy cancer cells. We then have also looked now at a set of genes that are involved in the inflammatory response, regnase and ROCN, that work post transcriptionally, really, to regulate a number of cytokine and inflammatory genes, and down through TCR signaling, but as well CAR signaling for regnase 1 and ROCN 1. And we did this data based on data from St. Jude that Hongbo Chi's lab has published now in a series of elegant studies where they identified, Hongbo found, using a CRISPR screen in CD8 mouse T cells, that regnase 1 deficiency really enhanced adoptive T cell therapy with TCR, transduced T cells shown here with OT1 or PMEL in a melanoma model, or with CAR T cells. That they work much better if in these syngeneic mouse models if regnase 1 is deleted. And so David, my lab has been looking at this and also knocked out the related gene, ROCN1, and now going back to our pancreatic cancer models, so with human T cells, finds much better control with double knockout T cells than with single knockouts, although regnase 1 is the best if we would pick a single knockout. So similar to what Hongbo has found. And we find unbelievable proliferation of these CAR T cells. These are mice that have over 1 million cells per microliter of the CAR T cells in the blood. And so the CAR T cells have much more proliferation and sustained anti-tumor effects with disruption of these inflammatory genes. And we think this would be interesting to take into a clinical trial in pancreatic cancer. Now, we've also looked at what we think are an orthogonal set of genes, the standard classic checkpoints PD-1 and CTLA-4. And this is unpublished work just submitted by my graduate student, Sonia Agarwal. So we went into this study thinking that because clinically, if you give antagonistic antibodies, IPI and Nivo, nivolumab, that in patients you get, in many studies, enhanced anti-tumor effects as well as enhanced toxicity when you have both checkpoints targeted. So we thought that by double knockout of both PD-1 and CTLA-4, we would get enhanced CAR T cell function. And my message is that's not true. What we find is single knockout of CTLA-4 is optimal. And that's what many studies that Sonia has, and I'm not showing it. We then went back to our CLL patients and took patients who had not responded to CAR T cells. I mentioned earlier there's about a 50% response rate in CLL to CD19 CARs. And we've taken here a non-responder where we had apheresed and saved upfront cells from the patient. If we have CTLA-4 knockout, the cells are able to clear a tumor, in this case a NELM6 CD19 tumor in NSG mice, whereas the cells of wild type CTLA-4 from not a healthy donor, but this leukemia donor are unable to clear the tumor. So there's T cell dysfunction in CLL T cells and CTLA-4 knockout ameliorates or abrogates that. Another approach is, and this is work we've done with Chris Garcia's lab at Stanford and Tony Ribas at Stanford, is to give CAR T cells along with adenoviral vectors. And as I'll show you, where we stumbled onto the fact that IL-9 signaling in CAR T cells makes them both more stem-like and more effector-like. So Miko Sirala, a postdoc in my lab, made mesothelin CAR T cells. Now in this case, it's targeting mouse mesothelin, the CAR, and it's mouse CD8 T cells that also express an IL-2 receptor and then switch receptors. So the signaling domain is either the bona fide IL-2 signaling domain or IL-7 or IL-9 signaling domain. And we expected to see more naive cells and perhaps less apoptosis with IL-7 switch receptor. And we didn't know what really to express with IL-9, what to expect. And what Miko found was that, it's in this paper that we recently published in Nature, is that if we signaled through the IL-2 signaling switch receptor and we expressed that with an adenoviral vector, so basically delivering a gene, and then added CAR T cells that had this switch receptor and signal to IL-9, that they had much more potent signaling, and that they had both preserved stem cell-like function, and it doesn't trigger the Tregs. So this appears to be a really attractive way to go, is to endow IL-9 signaling and CD8 CAR T cells. And, so we now have many tools, I think, to go for enhanced effects in solid cancers, many issues to overcome that are not present in a pre-B cell leukemia, where the primary mechanism of resistance is target loss. So there are many trials now, and we have Jim Riley, my colleague at Penn, using CAR T cells in HIV and AIDS, and that looks very potent to synthetically enhance the immune system. There are many trials with CAR T cells, CAR NK cells now, and they look potent. This is a study a few years ago now from Katie Resvani's group at MD Anderson, and with Sargil and Michael Klaczynski, one of my graduate students, he's made CAR macrophages. So using initially an adenoviral vector to express a CAR in a macrophage that then phagocytoses the tumor. And this is a trial open at Penn and also at NC State, UNC, with CAR macrophages with a targeted moiety being Herceptin, the anti-HER2, in solid cancer patients. So, and we think these are likely to be synergistic with CAR T cells, and in fact have studies underway to test that. And then we have had this really surprising finding that we can use CAR T cells to treat heart failure in mice. And I'll just finish with that. So the use of CAR T cells in autoimmunity and heart failure is a surprising turn. There's a case report from Erlangen about a year and a half ago now with a patient, a 20-year-old Vietnamese woman, treated with a CD19 CAR T cells and who had refractory lupus. Her double-stranded DNA antibodies went to 0, and she's now been in remission for more than a year George Scott has told me, and they've had this in many patients now in their phase 1 trial. So I think autoimmunity will be treated. We have 2 evolving trials at Penn. And then with Steve Alvelda, we've been targeting the stroma for many years with fibroblast activation CARs to target the CAFs, the carcinoma-associated fibroblasts that many people have shown in solid tumors are immunosuppressive, promote tumor growth, and block access of drugs and CAR T cells into the tumor microenvironment and sarcomas and uh, pancreatic cancer in particular. And so we made a FAP CAR against both murine and human FAP, fibroblast activation protein, and with a CD44BB signaling domain. And it's very active to kill FAP expressing fibroblasts when we use the FAP CAR T cells. And then we tested what about could it promote after cardiac stress or myocardial infarction, cardiac regeneration. And so we've given, and this is studies with John Epstein's lab at UPenn, these same CAR T cells to mice that have injured hearts. In this case, it's done through angiotensin-induced cardiac fibrosis and scarring. And we've looked into that because these are human specimens that are pathology group at Penn. This is a dilated cardiomyopathy staining for fibroblast activation protein, sarcoidosis, scleroderma, even COVID cases of cardiomyopathy have, as they repair these fibroblasts that impede the myogenic repair and promote scarring. So now we've treated these fat CAR T cells in mice that have failing hearts, shown that we can reverse the fibrosis, shown here with Pica-serious red staining the fibrosis, and a FAP CAR treated cell compared to controls CAR T cells in mice. And with Drew Weissman earlier this year, we tested, could we do this in vivo and actually use the same technology used by BioNTech, targeted lipid nanoparticles, to deliver the CARs in vivo to mice, and then could that repair CAR T cells? So in this case, the lipid nanoparticle has a anti-CD5 scFv on the outside, and it delivers a CAR to express CD19, and to anti-FAP. And this shows that we can do this in vivo in mice. The details are published in Science earlier this year. We could both express and get, without using a lentiviral vector or gamma retroviruses, but get in vivo CAR delivery with lipid nanoparticles and that they can repair the cardiac fibrosis. This shows end-diastolic volume in the mice, and all the details are in the patient paper in Science. So we're at this final point now where I think drug and the cell delivery for CAR T cells is forcing our medical care system to have now a fourth pillar, which was cell therapies, which is both in academia now and in the pharmaceutical industry. I've talked about the N of 1 issues for patient-specific manufacturing. I think in some cases they'll be manufactured in vivo. We've been working initially with Dr. Joe Biden to open a trial in Costa Rica, and I just had a signing ceremony with Steve Grupp in Costa Rica in May. And this is Dr. Biden, and the First Lady of Costa Rica is here. And we're going to be manufacturing CAR T cells in Philadelphia and treating kids with leukemia at the hospital in San Jose, Costa Rica, to see if we can establish this kind of therapy in a middle-income country where they do have healthcare routinely as a single-payer system. So this is an exciting project that we've started with Children's Hospital. And my closing message is we'll see many different kinds of combination therapy. And I know many of you are working on these at St. Jude to enhance the effects of cell therapies. And just like to close with this, many different uh, investigators and scientists and physicians that I've worked with at UPenn and Children's Hospital. Thank you for your attention.
Thank you very much, Carl. That was, truly fascinating. What an extraordinary story and history over the last, couple of decades in this field. It's so interesting to see all these different areas of activity spinning off from it. The basic biology is fascinating as well. So we do have some questions. Please keep them coming. Again, if you'd like to ask a question, please use the chat function in WebEx down the bottom right and address the questions to all panelists. So the first question is from Charlie Roberts. Might inhibiting IL-6 result in reduced efficacy of the anti-cancer effects of CAR T therapy?
Yeah, I mean, that was clearly our initial fear. And so we've, given, as I think you have there in standard of practice, basically the IL-6 is a rescue, the antagonism at the time in order to try to obviate the need to transfer into the ICU. So it was not given preemptively. Recently, Steve Grupp at Children's Hospital and the other colleagues have given it on to patients at high risk, so for CRS in a preempted fashion. And so far in ALL, it does not block the anti-tumor effects of the CAR T cells. I mean, of what we see clinically when you look at a month later at remission rates. So that's the data now. So first of all, there's some caveats to that. When we actually look at ALL and the cells that are making the most IL-6 are macrophages and not the CAR T cells. So when we looked with RNA using in situ hybridization, RNA-scope. So the CAR T cells are triggering macrophage activation and may be the ones causing the most damage, at least the immunopathology. And so ablating that, at least so far, has not impaired efficacy. But that doesn't mean that won't be true in other kinds of CAR T cells or other diseases, rather than in where we have the most data right now, which is pre-B cell ALL. But so far, the data is that in myeloma, and myeloma cells, as you know, also directly can secrete IL-6. So there may be an anti-tumor effect of giving the antagonist. And so far, there's the toxicity of CRS. There's not been any discernible difference in efficacy that I know of. So I think that there's need for ongoing studies. There's been proposals from several groups to block IL-1 rather than IL-6, and we'll have to see how that works out. It's certainly cheaper to use anakinra than it is to use tocilizumab.
Okay, right. So next question is, with regards to CAR T cells, what would be some reasons a treatment would fail after initially working for 14 days?
Sorry, can you repeat that again, Charles? Okay.
What would be some reasons for CAR T therapy failing after initially working for 14 days?
Yeah. So, so first of all, I think, unfortunately, it depends on the tumor and the tumor microenvironment. And a lot of you as cancer biologists know, I mean, the same tumor in a different organ can have different microenvironment. So a B cell tumor in the lymph node compared to the bone marrow has different resistance mechanisms. A lot of data that in KRAS tumors, that in the lung versus in the liver and a primary and a pancreas, there's different resistance mechanisms. So first of all, what happens at day 14 depends on where the tumor microenvironment is. The first resistance mechanism found was loss of tumor. Patient cells with CD19, that's a primary resistance mechanism, and pre-B cell ALL is evolution, a subclonal evolution to CD19 loss variants via several mechanisms. But in lymphomas, there's a lot of data from Kite with their CAR that, the tumor microenvironment in some is more immunosuppressive in some cases than others. Sometimes there's checkpoints implicated, and mechanisms really we don't know. And I was showing you in pancreatic cancer that, we find this NK T cell transition, which was a very surprising finding. Somehow, uh, the KRAS tumor cell can promote CAR T cells to become like NKT-like cells. And, you know, um, Mike Jensen and the group in Seattle published an extreme case of, and I'm sure you've seen this at St. Jude, and we've seen it as well now since, where the tumor can evade by reprogramming itself. And initially, a lymphoid tumor can become a myeloid-based tumor. And we've had one extreme example where an initial um, a mantle cell lymphoma that had classic CDK amplification, the tumor initially had a PET negative response, and at 6 months recurred with what our pathologist termed an undifferentiated pleomorphic sarcoma. And that tumor still had the initial CDK amplification that the parental mantle cell tumor had. So it had reprogrammed itself into a sarcoma. So what we're seeing with CAR T cells are resistance mechanisms that are pleomorphic, depend on the tumor type, and I think the organ, and then so it's a daunting task that we need to study, I think, to prevent these escapes from happening.
Okay. Question from Ben Youngblood. Can you comment on the persistence of CD8 positive TET2 knockout CAR T cells versus the enrichment of CD4 positive CAR T cells in your long haulers? What TET2 mediated program inhibits CD8 positive CAR T persistence?
So I wish, Ben, I knew the answer. There's we found, first of all, it's not just a one-off. When we've looked at 40 patients with you know repeated sampling in the patients, we found more than one patient that has insertion sites into TET2, and so that seems to be a favored site that gives clonal competition advantage of the CAR T cell compared to cells with wild type TET2. And we don't, there's a complexity to this. So I mentioned in our patient, it was a CD8 alpha beta T cell that outgrew. And then as Ben points out, what we found in our first two patients that didn't have the TET2 insertion, that it was CD4 cells long term. We don't know why that is. Michel Sadelain has looked, and I don't think it's published yet, but he's presented it, that in syngeneic mice with TET2 knockouts, he finds a CD8 lymphoproliferation like we do, but it's also the phenotype that's dependent on the signaling domain of the CAR T cell. So whether or not it has, CD28 signaling or 4-BB. So I think there's a lot of variables here that, that shape the phenotype of what we see in patients and what in the mouse models. What we know from clonal hematopoiesis is that in general, at the hematopoietic stem cell level, loss of function of TET2 gives clonal competition advantages. And as long as there aren't other genes like DNMT3A or AXL and so on, in general, that's safe in the hematopoietic stem cell. So there's a lot more to learn on to how we can enhance, I think, the function of these, effector T cells, and then at what levels they'll be safe and so on, and what do they need suicide genes and so on.
Okay. Maybe I can just ask one final question before we end this main session, and this is from Stephen Gottschalk. So can you comment how you're prioritizing clinical evaluation of second and third generation CAR Ts?
That's the problem, Stephen. It is because I think what we need rather than serial testing, which is what we're forced into in general by our FDA like paradigm of phase 1 trials, is we need multiple, there's so many, I showed many different edits that appear to promote the function of cells in solid tumors. Um, and it's in the animal models, they're not great as far as predicting what works best. So I am, so right now there's, you come down to what you can get approved by regulators and so on, and then and then the rapid pace of development in our basic science is far outstripping what we can tell and test in the clinic. And that's the fear. I think we have the tools to overcome all these resistance mechanisms, but the way to do it in patients is very painful, and as you know at this point, and expensive. And I think that we need to really, they do it more rapidly in China. That's the unfortunate truth, is they can do it more rapidly and translate cheaper than we can. And so right now, that's I think a real national problem we have of testing, these many different promising gene modifications and how to, and then combining. I mentioned, and I know there's work that you and others are doing, combining with vectors. Adenoviral vectors have many reasons why they might enhance CAR T cell functions. And then you have a lot of issues with the FDA on two different, gene modified vectors and so on. So there's the complexity of it translationally is slowing us down from our basic science advances.
Good points. So we've still got some questions, so please, if you'd like, stay with us for ongoing discussion, but we'll close the formal part of the session. And just to thank Professor Jude once more for a truly inspiring talk, a real tour de force. And just to mention, next week our own Ben Youngblood will be speaking, identifying epigenetic mechanisms of failure in T-cell based immunotherapy. So a somewhat related talk on one of the themes today. So thank you all again for attending, and we'll continue for another up to half an hour. Great. So we do have still a few more questions, and again, keep them coming if you'd like. So there are a couple of clinical questions that touch on a couple of themes you've mentioned. This is from Iram Eldam. Two questions. One of our patient, an infant ALL with MLL rearrangement, had conversion to AML after CAR T. What intervention can prevent this?
Yes, I mentioned, you've seen that. We've seen, that's been seen at Seattle Children's. So some of them are biphenotypic. This is something you guys know a lot more about than I do. So I think one thing we need is really careful characterization of the parental tumor and how subclonal is it at the beginning. But the FDA has been surprisingly flexible. So I think there is no problem with so-called polypharmacy, if you would here, giving CAR T cells or even TCR cells that target more than one lineage antigen. And so I think one way to prevent this is, it depends if you're gonna, an infantile AML, I think you're probably gonna want to go into a transplant setting, targeting both myeloid and lymphoid antigens at the beginning, I think is ethically fine because, we do see these escapes. And so I think that's where we need to head is it's really polypharmacy. I will tell you that I treated 3 patients at UCSF with a combination of CAR T cells that express CD19 and mesothelin. And we did that so that we could target IL-10 secreting, suppressive B cells, as well as the malignant tumor cell. And the FDA had no problem. We just had 2 INDs under the same clinical protocol. And I think you could do this. These infantile ALLs are awful in our outcomes, and I know you have the world's experience there. And the other main issue we found is, and I know, sure, is when do the T cells from infants really work? Because when they're first, the cord blood T cells don't work very well.
Is that right?
So you need to know when their T cells have matured far enough along that they're, useful, or do you have to use off-the-shelf third-party cells?
Do you think you utilize some of the manipulations you described in the second half of your talk to juice up the cord blood T cells, or what's
Yeah,
the
and
reason for that?
so some people have studied, it's initial, the cord blood T cells, when you activate through the TCR and the CD28, their cell division rate is great. It looks just like, naive cells from an adult, but they don't make interferon gamma.
Mm-hmm.
So they've uncoupled IL-2 path from interferon gamma. And I'm sure that's epigenetics. I haven't, that's solvable probably, and maybe some of the people on this know a lot more about that already. But I think, so I do think that's fixable to make cord blood cells better as effective. I mean, the NK cells work but the, alpha beta T cells, that's why we don't do, live viral vaccines in infants, as you all know. So that's so that's a cell intrinsic issue from the T cell side you need to look at in infantile ALL, as well as the issues of target escape and, plasticity of the tumor.
Okay. So another question is, so in relapse after CAR T, what are the treatment options in leukemia? Can we give another CAR T?
Yeah, and that's so in general, the FDA has not had a problem with that. The results have not been good so far, if by talking to people. In some cases, you can show that the patient has the induction of anti-CAR responses. So FMC63 is a mouse hybridoma, and you can show that the patient has responses immunologically or reject new kinds of CARs. But we'll show at ASH. We've had some good results now in CAR refractory patients to CD19 CARs, where the CAR T cell now makes IL-18, and so we're seeing responses there, and so somewhat it depends on, did the patient relapse from previous CAR cells due to target loss, or was it due to rejection of the incoming CAR T cells? And I think the approach is going to differ.
So another question's a little related to that, and resistance or failure of therapy. So let's just jot. So for patients who have been in remission but cancer returned, is there a suggestion that the phenotypic shift of CAR T cells goes in the other direction that has been shown for those with greater than 10 years in remission?
Yeah, we don't know this mechanism of, if you look at classic immunology, all the textbooks say that, if you give, say, ovalbumin to a mouse, so a foreign protein, they'll make a CD8 cell, of which the prototypic OT1, but they'll make CD4 CTLs and helper T cells. And then if you look at the memory cells long term, they're mostly CD8. And so that's how the usual human immune response is. And with CAR T cells, our results are, there's, first of all, it's shaped by how you make a memory CAR T cell. We don't know. I mean, they dominantly see, I think, mostly blasts, which don't have the normal amount of co-stimulatory molecules on them like a dendritic cell. And then they'll see some normal B cells, and then later the blasts are cleared. And what they see that has antigen long term is these pre-B cells coming out of stem cells. So that's not the same as a sterile immunity after a viral infection, where the antigen would go away. So the CAR T cells, it's first of all, the propensity to make a memory cell we found is much more if they have 4-1BB than if it's CD28. If it's CD28, they're all gone within a month. And with 4-1BB, they persist, and we found this unexpected outgrowth of gamma delta cells. And that we don't know if there's ligands on the blast cells, that promoted that or why that occurs. And so the mouse models are not very good at studying this. That's, so we need to study patients more. I think myeloma will help get us beyond, is it something to a CD19 pre-B cell tumor that shapes what memory CAR cells look like, or is it, um, due to, uh, the target with a plasma cell, for instance. Now, when Marcella Mouse was at Penn, we studied 15 patients with glioblastoma, giving CAR T cells against EGFR variant 3. So, a tumor neoantigen that's a splice variant. And there we found, tumor loss in about, isoform loss of the target antigen in about half the patients. And in solid tumors, and I know you have a lot of this, like Steve Gottschalk there, a lot of experience in solid tumors. Mostly the CAR T cells leave the blood pool and go into the tumor microenvironment. And then whether they survive or not long term probably depends on many complex issues of is there helping function and so on. And, is there this issue we see with the KRAS tumors of plasticity and then K cell outgrowth? So there's a lot of issues now to find out what would be an ideal long-term memory cell in a solid tumor and where they don't have the blood pool and the issue of ongoing vaccination like we have with a CD19 CAR cell.
Yeah.
So it's a fascinating area.
We've got a couple more if we can. So what's the mechanism of CAR T cell induced cell death against target cancer cells? Does IL-2 and or IL-9 induce transcriptional responses in the CAR T cell affect this cell death mechanism?
Yeah, so I haven't studied that in great detail. There's studies. So IL-9, I think, in part may circumvent some of it and answer it, whereas without, so that's, I think, but we don't have great data on that, I think. And then Nathan Singh, who's now at WashU, has studied and he's found both that death receptors in on both the T cell side, and Marcella Mouse had a paper in Nature looking at this, and then Marco Aurela at Penn. It seems to depend if it's a solid tumor, the mechanism of cell death of the T cell is different than, and escape mechanisms are different than if it's a liquid tumor, meaning a bone marrow derived tumor, where integrins are very important on the initial, and so it's a complex issue, and you can't just say one answer of a CAR T cell and cell death. It depends if it's targeting a solid tumor, a liquid tumor, and then what the signaling domain is, and all. There's many escape functions, unfortunately.
Okay. Another question from Charlie Roberts. Are there genes other than TET2 in which there are recurrent insertions?
Yeah, so we have some of those in a paper we had in JCI, and clinic, and one of them is for sure CBL-B. And that was reported from Kotchenterfer's group at the NCI, and we've seen the same. So CBL-B, is a scaffold molecule that prevents and limits signaling through CD28, and so CBL-B loss of function can promote CAR T cell function in vitro, and that looks to be like a good way to go as well. So I think, and then there's other genes. I think Steve Gottschalk's seen DNMT3A as another way, which is, so epigenetics are a very potent way, I think, as targets to promote CAR T cell, either delay this dysfunction or to prevent it. So that's an exciting way to go, and the questions are how to do it safely and all that.
A question from Steve Harrington. I'd love to hear your thoughts on the potential for biomaterials such as hydrogels for enhancement of CAR T cell therapy in solid tumors.
Yeah, so I mean, I think, can we make a way to boost the cells and promote their function. And so that's so nano engineering biomaterials look like a great way to go. And one of Steve Gottschalk's former colleagues, Petro Doty, his group now at UNC has shown in glioblastoma that, using a hydrogel, which is fibrin sealant. So basically fibrin and thrombin, if you mix CAR T cells into that in a glioblastoma cavity, the cells work better. And we now have found the same thing in resected peripheral tumors. So I think fibrin sealants are a good way to promote function and engraftment of cells locally, and it seems to improve their therapeutic index. And I think there's many advanced ways with biomaterials to do this, and I think we're going to see a lot of innovation in that area.
Okay. I'd like to ask a question, if I can, come back to BALL. What are you thinking about bringing the therapy earlier? We've had the discussions here, and the issues come up in terms of the logistics, the engineering, through to the potential toxicity, perhaps accepting that you're backing away from curative but prolonged chemotherapy. What are your thoughts in terms of perhaps using it as an initial therapy rather than a relapse?
Well, first of all, I think the patients count the most, and the patients for sure would rather have it initially, and a lot of feedback we've had. I think, the main, and there's a lot of what St. Jude's done, getting rid of cranial irradiation is a huge issue, if you have a kid. And so that's any way that goes to get rid of that is going to be, I think, beneficial. Now, we now are just opened a trial where we're giving cells, we can apheresis, and the cells are ready for infusion 3 days later. So one of the problems right now with an upfront therapy is, what do you do to bridge or not? And do you give some holding chemotherapy, and do you need lymphodepletion? So if we can get around lymphodepletion, so one way is, can we get in pre-B cell ALL, get a way where it's completely chemotherapy free and just give some forms of cell therapies, CAR T cells, maybe recombinant proteins, BiTEs, or whatever, because the BiTEs could be off the shelf in there while you're manufacturing autologous cells. And so I think this is, we need these trials to be done. And I don't know, do you initially pick people with bad karyotypes as the ones most likely that, they would relapse, and then they have a crossover design where they could be given CAR T cells or conventional therapy and crossover. This kind of thing that it's not my expertise, but I think we now have enough data, the safety. I was most worried with CAR T cells initially of the so-called Frankenstein cell, where the CAR T cell would have transformation or something. So that hasn't happened now. It's much safer than chemo. I think that we have really good data. The other issue is that I thought was a risk, when you give activated T cells, maybe we would cause autoimmune disease. Because that's activated CD4 and CD8 cells lead to lupus, they lead to MS and stuff. We haven't seen that in our kids. And then what we have had as a predictable some incidents of B cell aplasia. And that's turned out to be to be very manageable. So I think we now have a good enough data set to promote or propose therapies up front. And then it comes down to the issues of how, the vein to vein time, how would you do it in practice, who would you select, and, as a demonstration for this. And, so I think it's ethical. And certainly, if I had a child right now, I would do that, rather than 3 years of intensive chemotherapy, you all know what that is. So,
And perhaps related to that, the issue of whether it's a bridge to transplant or not. So do you think that giving it earlier will change that, that dimension at all?
Yeah, well, yeah, and I think clearly a transplant can be a backup, and it depends. Infantile, the different is different than you have an 8-year-old that has sort of classic pre-B cell ALL. I mean, we found because of standard of care CHOP, we didn't cross the patients over into transplant, so we now have a whole large cohort where it's been a standalone therapy. Now we do monitor them for, early recurrence, loss of B cell aplasia, because then they're at higher risk of relapse. So I think if it needs to be done with careful tumor monitoring and do they have functional CARs or not? And then there's this issue of, B cell of CD19 loss. That clearly is going to go away with like the approaches they have at Stanford with a CD19 22 dual CAR or CD19, CD20, et cetera. I think that'll get rid of the tumor, hopefully the lineage loss issue of targets. So I think those things we need to get in place. And then eventually we'll be able to have, I think, an approach where we don't require allogeneic stem cells, but hopefully, next generation CAR T cells might be sufficient in many patients.
Well, great. I think we've gone through all the questions. So, thank you again. This is really fascinating. It's amazing to see what you and the group are doing. It's really incredible
Well,
stuff.
I think the whole field it's really exciting to see what we have on the way. So thanks so much for inviting me, Charles. And it's great to be with you.
The extended time is much appreciated. So thanks again.
Yeah, bye-bye.
All the best. Bye.